Welcome everyone to this lunchtime discussion of the history, science, and art of Lewis and Clark's Herbarium. Before we get started, I'd like to start with a land acknowledgement. The Museum for Art and Wood is situated on the traditional and unceded lands of the Lenape and Wingo-Hawking. In our acknowledgement of their continued presence in their homeland, we affirm the aspiration of the great Lenape chief, Tam and that there be harmony between the indigenous people of this land and the descendants of the immigrants to this land. As long as the rivers and creeks flow and the sun, moon, and stars shine. We also hold gratitude for the network of plants, animals, soil, water that evolved over millennia and compose the ecosystem that supports the current exhibition and study, as well as the core of the museum's mission. Again, welcome. I'm Katie Sorensen, Director of Outreach and Communications. And today we have a really wonderful talk for you, a collaborative talk with the American Philosophical Society, the Academy of Natural Sciences, and the Museum for Art and Wood. We also have today from our current exhibition, To Understand a Tree, artist Gina Siepel, who will be participating in today's talk. And with that, I'd like to go ahead and introduce our wonderful participants today. Allie Rosen is Education Programs Manager at the American Philosophical Society's Library and Museum. Allie creates, coordinates, and implements intergenerational school outreach and informal adult programming at the Society. She also has worked as an educator at the Independent Seaport Museum and holds an MA in Museum Education from the University of Arts and a BA in History and Political Science from Rutgers University. Welcome. For our next, next slide I should have on. For our next participant is Dr. Rick McCourt, who's been a botany curator at the Academy of Natural Sciences of Drexel University since 1996 and a professor of the Biodiversity, Earth, and Environmental Sciences department since its founding in Drexel University in 2012. So that's amazing. And Professor McCourt's research program targets the biodiversity, evolution, ecology, and systematics of green algae, specifically green algae that are among the closest living algal relatives of land plants. In addition, he curates the plant collection of Lewis and Clark expedition and has written about the history of the botanical results from their journey. Welcome, Rick. And then Gina, who we know very well here at the museum, is an interdisciplinary artist, designer, and woodworker based in Greenfield, Massachusetts. The Pocumtuck, did I say that, Gina? Or you can correct me, okay. Land. Their artistic practice reflects an engagement with place, history, queer experience, and ecology. And their work integrates conceptual concerns and craftsmanship with a focus on wood as a natural and cultural material. Gina's works have been shown in museums and galleries nationally. She is currently a McLeish Field Station Artist in Residence at Smith College and a 2023 recipient of a Teaching Artist Cohort Grant from the Center of Craft. So welcome everyone. This is very exciting to have you all here today. And I'm gonna go ahead and hand this over to Allie, who is going to start us off.
Cool. Thanks, Katie. I'm gonna share my screen. Cool. Wonderful. So, hi everyone. Like Katie mentioned, I'm Allie. I'm from the American Philosophical Society. And I'm gonna tell you a brief history on how these plant specimens and later that would turn into herbarium sheets got here to Philadelphia. Now, your first question might be, what is the American Philosophical Society, and what does it have to do with plants and herbariums? The American Philosophical Society was founded in 1743 by none other than Benjamin Franklin to promote useful knowledge. And when we say useful knowledge, Franklin wanted people, if you have a great idea, not sit there in an armchair and be like, I have this wonderful idea, not do anything with it. He wanted you to go out and have a positive impact on society with that idea. Now, you might be wondering, what does philosophy have to do with this? So when we were founded in the 1700s, philosophy meant more natural philosophy, what we consider science today. So our members, who are not your typical, they're not your museum members, they're members who are at the top of their fields. Think of them as scientists back then. Today, the APS, and that's our building you see there to the left there, that's it on 5th Street between Walnut and Chestnut. Today, the APS is still a functioning society. So like I mentioned, our members, they meet actually twice a year in April and November. We also have a research library, which houses around 14 million manuscripts. And then also a museum, which this is the building where our museum's at, houses a rotating exhibition every year. Before I get into the talk, I'll do a quick thing about the exhibition, which is currently Sketching Splendor, American Natural History, 1750 to 1850. And this exhibition focuses on how through words and images, William Bartram, Titian Ramsay Peale, and John James Audubon captures nature's splendor and engaged in a variety of scientific approaches. But of course, it wasn't just these men working in this field at this time. They were also relying on the knowledge, expertise, and labor of the indigenous enslaved people as well. So if you're in the area in Philadelphia and want to see a show about natural history, come on down. We're open Thursday through Sunday, 10 to 5. But let's backtrack now. Let's go a little bit farther into the past and talk about how these plant specimens got here to Philadelphia. And like many stories in the APS, it starts with Thomas Jefferson. Thomas Jefferson, he was the president of the APS. He was elected to the APS in 1780. He was president from 1797 to 1814, and he was always interested in science and always had his hand in a lot of different scientific ventures, whether it be from a systematic weather data collection, to investigating fossils, to finally wanting to send expeditions out west to collect natural specimens, whether animals, plants, geography, and all that. He was always interested in this. And early on, he was interested in sending an expedition out west for both scientific and political reasons. Now, his first attempt to go out west was with a French botanist named André Michaux. And think of him as Lewis and Clark 1.0. He sent Michaux out there with pretty much pretty similar goals to Lewis and Clark, finding a passage out west, collecting information on plants and animals and bringing back those species, engaging in talks with the indigenous people out there. So sounds pretty matter of fact, right? Unfortunately, Michaux got into some trouble with some international politics with Citizen Genet and some spying. So unfortunately, this expedition didn't get very far before it was canceled. So it takes until the Louisiana Purchase, and in 1803, Jefferson nominates puts forth his secretary, Meriwether Lewis, to be the head of this expedition west. That was to go west, chart your territory, make it to the Pacific, collect plant specimens, animal specimens, document the whole passage out there. But to do that, Meriwether Lewis needs to also learn a few more things before he goes out west. He needs to get his supplies. So as I like to say at times, Lewis and Clark kind of started in Philadelphia. So Jefferson brings Lewis here to Philly, to Pennsylvania, to learn from APS members. And again, these are people in the top of their field. And so you'll see here, these are just some of the members that Lewis met with, whether it was with Robert Patterson and Andrew Ellicott. He was learning about surveying and astronomy, whether with Caspar Wister here, he was learning about anatomy and fossils. Lewis also met with Benjamin Rush, where he received medicinal supplies and also just learned about basic medicine. It was here too, he received thunderclappers, which were these mercury-laced pills, which, you needed, back then, if they thought there was an imbalance, you had to get rid of it. So They were in times would take these mercury-laced pills called thunderclappers. And finally, he met with Benjamin Smith Barton here in the corner, where he learned about botany. While Lewis had a rudimentary knowledge, he also learned from his mother as well. It was through Barton that he was going to learn further more about how to collect, how to preserve these plants. And so they head off here into their expedition. So it goes from 1804 to 1806. This is a map of their expedition west. So again, their goals were make it to Pacific, document as you go the animals. So this is as they were culminating on their expedition, starting on May 14th, 1804, arriving at the Oregon coast in November 1805, and returning east and arriving back in September 1806. They documented this expedition in a series of journals, as well as collecting these live specimens. So this journal, you can see a California condor they saw, identified, and later wrote in the journal, also as well as this white salmon trout. They also had to map the territory. So you can see here, Clark was more of the cartographer, so he was drawing these maps inside the journal. They were also noting the weather, right? If they're collecting these plant specimens, they need to know and also where these animals live. And if they want people also to expand west, they need to know what the climate and weather's like. And then down here, you can see a little leaf, but we want to see the actual specimens, right? That's just a tiny little leaf. So these here would later be the herbarium sheets that Lewis and Clark would collect. So these would be the dry specimens that they would collect. And Lewis would collect hundreds of these plant specimens throughout his travel. So it's believed that they possibly pressed the plants, attached them to sheets of paper, and Lewis would write down where he found them and then wrap them in oilskins to protect them along their travels. It's these plant specimens that then would get sent back to Jefferson, right? And then he would send them along to the American Philosophical Society, which was acting at the time as a depository for these natural specimens. Besides the APS, that building I showed you was built in 1789, and at one point the Peel Museum was there, which was the first successful public museum here in Philadelphia. So a lot of animal specimens got sent to Peel, while the plant specimens got sent to the APS. And this would culminate to today, where the majority of these plant specimens that would later turn into these herbarium sheets are in the possessions of the American Philosophical Society, and then the Academy of Natural Sciences also has these herbarium sheets as well. So the majority are with either both these organizations, and then there's like around 9 or 10, I think, that are in England. And that's a story for another time. But these do tell a story. There are some of these sheets do have still some of Lewis's handwritings on them that you see in these small fragments at the bottom here. But to tell you more about these herbarium sheets and the science behind it all, I'm going to pass it off to the expert here, to Rick. So Rick, here you go. I'll stop sharing.
Thanks. It told me to unmute myself. I managed to go ahead into the talk here a little bit, but I'm going to. I hope I don't repeat too much of what Katie said there, but I'll go through a few things. This is what's called Traveler's Rest, a place where Lewis and Clark rested. They needed a lot of rest on the way, and I picked it because it's a nice place to see. A couple of things about the expedition. First of all, this is what I'm going to talk about today: botany in 19th and 18th century America, plant exploration, and then collecting and preserving Lewis's dry garden, his herbarium specimens. This is Traveler's Rest in 2004 when I was there. A picture by someone else, but this is what the plant looks like, called bitterroot out in the wild. It was
Rick.
Yes.
Rick, can you share your screen?
Oh, I'm sorry.
No worries. That's what we're here for.
Yeah. Hang on a second.
No worries.
Oh, there we go. Yep, there we go. Sorry about that. The buttons disappear when I turn it into one of the
there we go.
preparation.
Okay, so now we're seeing.
Yeah, here we go. So here is what Whoops. Sorry about that. Here's what bitterroot looks like in the wild. And Lewis, when he found this, did what every good botanist should do. You collect it and press it flat and dry it out. And so this is what the herbarium specimen looks like that came from that beautiful plant. All that's left are the petals, because the root was used to try to revive it and rejuvenate it by growing it back here in Philadelphia. I think that was successful, but they didn't save the root. And so that's why it's called Lewisia rediviva, which means Lewisia resurrected. And in a way, the plants really resurrect Lewis's legacy for us. Here's the expedition map, goes from St. Louis out to the Pacific coast and back. But of course, we know that it really started out from the East Coast all the way to the West Coast and back. The states were not there, but just when they set out, the Louisiana Purchase was completed by treaty, and so they were exploring Western America, quote unquote, from their boats. They started out in the East, roughly here in Monticello in Jefferson's library. And I've done a little photoshopping there, and you can see the Rocky Mountains out of his window in the back, which he couldn't see, but he could explore them through his library. And that's why he was interested to send Lewis out there. This quotation from Jefferson is one of my favorites. The greatest service which can be rendered to any country is to find a useful plant and add it to its culture. This is a lot of why he sent Lewis out to get the plants and collect them. He sent them, Lewis, to Philadelphia in 1803, as you heard just a moment ago, with these instructions that he took with him on the journey to go and find out about the growth and vegetable productions of the United States. And in order to do that, he did need a bit more of the technical training that he didn't quite have, and so he consulted Benjamin Smith Barton. This is a slightly different portrait of the one that you saw earlier, but he met Barton in Philadelphia, who lived right downtown in just a small few blocks of Old City area. And it's not written about in specifics, but we know from sort of the forensic evidence left behind that he probably learned to press plants from Benjamin Smith Barton, who knew how to do this. This is a book, or pages from a book by Barton, assembled in 1795. This is a specimen of a plant from Bartram's garden. You can maybe make that out on the lower right there of the picture, where it's pressed inside of a book with a blotting paper on the left and a regular white herbarium sheet on the right, not the standard size we use today, which comes in to be very interesting later on in the story. But this is how they were pressed, and Barton showed Lewis 2 volumes of this, and probably showed him this is how you press the plants, this is how you get them to be preserved for posterity. Lewis also drew a lot of little pictures in the margins, but that wasn't going to be enough. Drawings and descriptions would not have been enough, or would not have been enough in my spell check there. So he was directed to collect the plants and make what's called an herbarium. This is the cover of a book by Barbara Tiers, who is of the New York Botanical Garden. It's a great book, beautiful book, and Lewis and Clark is mentioned in it, but also the other people who were responsible for our preserving plants for scientific study. And these were called horticicas, which translates to dry garden. And this is the gentleman who managed to do that for us. His name is Luca Quina. You can't see the top of his name there, but he lived in the late 15th century to early 16th century. And he got the idea of putting the books in, or plants in books. His idea was, how do I show medical students about the plants they're going to use to get medicines from and cure their patients when the plants die off in the winter? And so he developed the idea of pressing them and keeping them preserved. And so you press a plant on a dry book page, you glue it to the page later, and voila, or in Italian, ecco, the Hortus Himalis, or winter garden, or Horticicus dry garden is there for you to study. A little while later in the 1700s, George Clifford got the idea that maybe we should take those books and take them apart. Then you could put things on shelves and keep them in cabinets for better organization. And the gentleman on the right there, Carolus Linnaeus, adopted this technique, and it's now how we do these things. So what did it look like when Lewis was pressing these plants? Well, we have this heretofore never been seen picture of Lewis pressing a plant. Just kidding, this is me with a plant in Philadelphia with a newspaper, for those of you too young to know what that is, in which is perfectly sized to press a plant, put it inside of a plant press with blotting papers and absorptive material, covering O2 boards and pressing them flat to express all the dry matter and material. After it's pressed, it is not really quite ready for the primetime museum, but for that you use good quality, high rag content paper, archival glue, maybe thread and needles, little strips of paper to put on top of the plant, and you get a specimen. Now that takes a while in Philadelphia. It probably took longer for Lewis, who's shown here on the right with Sacagawea talking to other Native Americans in the boats. I've scoured this photo. I don't see a picture of a plant press, but maybe it's in the lower right there. We don't know. It's really not known how they kept the plants dry, but they had to be dry or else we wouldn't have them today, which is quite fascinating. This is what the whole collection looks like, kind of spread out and photoshopped into one poster. The plants have lost their color, but they still have their shape and morphology to a great extent. They've lost a few little pieces here and there, but we keep those pieces when we can find them in little fragment packets on the side. This is an individual sheet shown. It has the Academy of Natural Sciences little stamp on it. That colored photo of Lewisia and Clarkia, our little imprint there, is not on the sheet itself. It's just an image here. But on this, you can see not only the plant specimens themselves of an Artemisia or sagebrush type plant, but you see little annotations by scientists. It's not quite graffiti, but it's technical graffiti in the sense that they're annotations with interesting information about the plants, re-identifications or confirmations of identification by scientists later. But at the lower section there, you'll see a purple piece of paper. That's Lewis's handwriting himself. And so on 34 of the specimen sheets, all from the American Philosophical Society, we have those. The rest of them were probably thrown away. One was found at a yard sale or at some antique shop, so you might keep your eye open in Philadelphia for this kind of thing. But we don't know what happened to the rest of them. They were probably just discarded as ephemera. You'll notice that the blotting paper that Lewis wrote on kind of is about as wide as the specimen there. That's a coincidence in that case, but that actually has probably some validity to it. It's roughly the same kind of texture of the paper that Barton showed Lewis. And so Earl Spalmer, who's actually listening today, can correct me on anything I get wrong, deciphered the mystery of this, which is probably that Lewis probably pressed these plants inside of purple blotting paper like this and kept them that way. And one little evidence of this can be seen on this two specimens. These are two specimens that were treated differently. On the left is one that's at the American Philosophical Society. On the right is one that went to London. Stay tuned for how that happened, but I won't go into great detail today. The one on the left was never unfolded. The one on the right was, and you can see fold lines in it. And so it looks a little bit better. But the one on the left was probably the dimensions of the press or the blotting paper that Lewis used to preserve these plants. And if you overlay this here, you see I kind of resized it so that it's a little bit of a fake news type thing, but it probably looks something like this, and the blotting paper was about that size. Back to Philadelphia, the plants came with Lewis. It's a bit of a convoluted story, but working at the Woodlands, which is now a cemetery here in Philadelphia, was a beautiful botanical garden at the time, probably the most grandiose in the United States of that era, was Frederick Persh, a gardener who helped Lewis sort out these botanical discoveries. He went through all these, he made annotations on them, gave Lewis the notes, and then left town. Left behind were the plant specimens, which stayed with Benjamin Smith Barton, went to the APS and were stored away there. And for about 80 years, nobody really paid all that much attention to them. Meanwhile, Persh, who had left town after working with Lewis, went to London, and without telling anyone, he had taken 57 plants with him to work with this gentleman on the screen here, who had an herbarium of about 50 to 60,000 specimens. Working in his library and with his specimens, Persh wrote a book. This was about 1813, the middle of the War of 1812. He went to London probably to get away from the ruckus going on in New York during the War of 1812 at the time. But he wrote this book called Flora of North America in Latin. And it was many of Lewis's specimens were cited in it, and he gave him credit for it. So that's kind of nice. He was powered by beer, they say, but we don't know for sure exactly the role it played, but he did like beer. The plants were auctioned off when Lambert died, and a fellow named Tuckerman bought them, or bought 47 of them anyway, and brought them back to Amherst College. And he kept them for about 12 years, at which point he sent a letter to the Academy of Natural Sciences and said, maybe you want these specimens, take good care of them, and I'll come down, and if you'll let me look at your lichens, I'd appreciate that. So when were the specimens mounted? This was a bit of a mystery until not too long ago, within our lifetimes, basically. This is how they were, an end-on view of the specimens looks. But in 1982, John M. Fogg, just a few weeks before he passed away, reminisced that Francis Pinnell, a curator at the Academy of Natural Sciences, supervised his first task in the herbarium of 1921, mounting the APS specimens. And so that means they were probably loose there for about 25 years. Why you would give this as the first task to a person starting out his career is not known, but he did a good job, luckily for us. These folders are where the plant stayed until 2002, when Earl and I got a Saving America's Treasures grant, and we, in consultation with the Conservation Center for Art and Historic Artifacts, developed some new holders for them so that we could more safely display them and keep them for posterity. We have new cabinets and everything else, so they're really kept in good shape now. So they're in the Types and Special Collections room. Here's one of them being held up there. So here's a picture of Earl. He'll be embarrassed I show it, but he's my co-conspirator in all this. And like I said, he can correct me if I've missed anything. I want to dedicate this little talk to Jim Reveal and Joe Musselman, two Lewis and Clark scholars who helped me along the way. And I'll finish with this picture of Lewissia and Clarkia and the Lewis and Clark Herbarium.
Oh, thank you so much, Rick. That was amazing. And let's see. Great. And Gina.
Okay, let me just share my screen. All right, can everybody see that?
Yes,
Yes.
it looks great.
Good, good. So it's so nice to hear about the Lewis and Clark Herbarium and to just think about the sort of tradition that the work that's part of my show, To Understand a Tree at the Museum for Art and Wood, connects to. Rick, I particularly love that little Amherst College connection at the end, because my collaborator on the herbarium, naturalist and botanist Kate Wellspring, and I met at Amherst College working on a project with taxidermied birds that belong to John James Audubon. So really nice connection there. So the basic kind of fundamental question of the work in the exhibit, To Understand a Tree, is really a question of relating to and kind of coming into a deeper understanding of a particular tree and of the forest from the perspective of a woodworker. I began with a question about a tree, and I very quickly realized that to try to relate to or understand a tree is really, in some ways, a kind of misunderstanding of the forest, and that there's really almost no separation between the forest and the tree. A single tree in the forest is just an entry point into a complex and dynamic ecosystemic community. The herbarium is a kind of, that's in the show, is a kind of representation of that. This herbarium is a result of a collaboration between myself, and I'm an artist and a woodworker, and Kate Wellspring, who is a naturalist with a strong focus on plants and botany. And so, there were several people who worked on this herbarium, but I'm going to kind of try to just represent its role in our work. Whoops. So, our project focuses on this single red oak tree. It is about 100 years old. The species is a northern red oak. It is in the forest at the McLeish Field Station of Smith College, which is in the sort of northwestern part of Massachusetts and kind of the foothills of the Berkshires. It's in a pretty dense part of the forest. There's several thousand acres of continuous forest there. We're focusing on this tree because red oak is a sort of classic, furniture wood, and also it's like a keystone species in the ecosystem here in Massachusetts. This tree is about 100 years old. It's about 85 feet tall, And in order to learn more about this tree, we defined a site. We took the height of the tree, which is 85 feet. We put an 85-foot rope around it, and we made a big circle in the woods. So we got this big kind of 170-foot diameter site. So that site right around the tree is where the herbarium specimens came from. What you're seeing here is an image of a video still of a durational video shot over the course of an entire year that you can see in the exhibition at the Museum for Art and Wood. This is just one still image from that video, and it kind of is a nice representation of what the site looked like in this. I believe this picture was from the summer of 2019. So here's the herbarium in the exhibit, along with some other works, some of which are drawn from the forest also, like the carbon study forest floor on the lower right, and also some of the sculptural chairs. And you'll see there's 2 panels, and then there are field notes of an artist, which are a little compilation of notes that I made through the time at the site, and also field notes of a naturalist, which are some of Kate's notes. And I'm just going to show a couple of images of the herbarium. So this is the sort of back wall. I think we have about 22 specimens, or 23 total in the exhibit, all drawn from the site. And there's the other wall panel. Here's a close-up of a few of the images, and it's really interesting to see in the Lewis and Clark herbarium the color, how much the color has faded, because you know, our specimens were collected between 2020 and 2023, so they're still pretty new, and they still have like a lot of chlorophyll. So that's kind of interesting to see how much they are likely to fade. So there are several micro communities at the site, and this is how Kate chose to select the plants that are in the herbarium. I really wasn't part of that. That was kind of one of her big contributions to the project. The site has a lot of diversity. It's part of why we chose that area. So there's the sort of area around the red oak, which has some large mature trees like black cherry and white pine, and then some saprophytic plants that coexist with those trees like pine sap and ghost pipe. There's the trail in vicinity where there's more sun, more wildflowers. The big tooth aspen is from there. There's an area around the old stone wall, which is very typical in New England. And so certain plants like that habitat. There's a vernal pool on our site. And then there's a bog and a stream connected to that. So there's a lot of ferns and a lot of plants that like water. Near that, there's another kind of southeast side of the trail that's a little more open and sunny, and then there's the stream bed. So the plants are organized according to these kind of micro communities at the site. And Kate's notes kind of show you a little bit, like if you read Kate's notes, if you go to the exhibit, her notes will kind of walk you through the specimens and how they relate to each other in this kind of ecosystemic community. This, on the left in this slide, you see her notes from the boggy area, and then I put in an image on the right of what the boggy area actually looks like in the summer when there's been a lot of rain. And I think that part of, for me, what's valuable about the herbarium is it gives you a chance to kind of pull out, you know, zoom out a little bit from the like density and the confusion and the, you know, layering of the actual natural environment, and you can kind of appreciate an individual plant. So it's like an act of extracting something, you know, it's like a figure ground relationship, to use an art term, like taking, you know, this subject in isolation and being able to see it, which is really different from how you experience it in the ecosystem. One of the things that I think is really important about the herbarium, at least for our process, is that it becomes a snapshot of the forest at a particular time. These specimens were selected over about 3 years. And, you know, it's not comprehensive. I mean, we're not doing formal science. And so we have some freedom to be sort of subjective and kind of just make choices about what we're including. But I'm very interested in how much the forest is changing. We've seen a lot of change at the site just in the 5 years that we've been observing it. So this is a moment, and it's going to be different in 10 or 20 years. So these are just a few things that I see happening at the site. Presence of invasive species on the left, that's the sign of the hemlock woolly adelgid. A huge drought in 2022 that really affected plant life at the site. storm damage in 2023 and 2024 over the winter, taking trees at the site down, opening up a lot of light, that will bring a lot more plant life in. And so this site is really changing, as is everything. And interestingly, with climate change, they say that depending on what happens and how it's dealt with, the climate of Massachusetts will become, best case scenario, more like the climate of New Jersey or perhaps Philadelphia, and worst case scenario, more like the climate of North or South Carolina. And so that's a very different plant community that we'd be looking at. And I think that, to keep herbarium specimens like this just is very valuable in terms of just documentation of these changes. This is a studio assistant that worked with me, Nadia Mogilevsky, who's a Hampshire College senior art major. And she helped me work on mounting the herbarium specimens. Kate taught us how to do it because Kate has expertise in this from some of her work with botanical organizations. She taught us how to do it, and then Nadia and I worked together to mount the specimens. So you can see we have a sheet of plexiglass coated with archival glue. We're using the archival paper, getting the glue on there in this particular way, and then kind of using tweezers and arranging them. We did make kind of like aesthetic choices in how we mounted the specimens, but we also tried to stay true to the growth habits of the plants in their environment. And then lastly, I'll just finish with this image, which is a piece called Living Material 1, Red Oak Seedling. This is a red oak seedling that is the offspring of our tree that is the focus of our project, which is, from the video still at the beginning, the 100-year-old red oak tree. So this is one of the seedlings. And this is part of a series of works, Living Material 1, 2, and 3, that kind of metaphorically and poetically describe or reflect on the life cycle of a tree, beginning with birth and ending with death. So this is kind of taking a herbarium specimen and actually using it in a poetic dimension as well as in a sort of documentary kind of dimension. So all of these kinds of impulses are present in the show. And I think I will stop there and pass it back to Katie.
Wow, this is so wonderful. Thank you, Gina, Rick, and Allie, for sharing with us. I feel like herbaria is not talked about enough, and I think it's something that is really accessible to all of us to do on our own and explore the nature around us, even in our own backyards. So I'd like to open it up to questions from the group. And this is a small group. If you'd like to just go ahead and unmute yourself, feel free to. If you're more comfortable with putting your question in the chat, you can do that as well, or raise your hand.
I have a question. I'd like to ask I'll get Gina's comments on this anyway. You notice that the specimens seem to be sometimes a bit haphazard on the pages that I showed, because sometimes they sort of are. But when they're pressed, they're definitely some workers in the herbarium we see, like a fellow named Byard Long, who was a curator at the herbarium over half century ago, who made beautiful specimens. He laid them out in certain ways that really were aesthetically pleasing. I wonder, have you tried to do that, or have you attempted to take that into account with what you do?
Yeah, I love thinking about that. And I did a lot of research on what do these things look like before we started, how are they displayed? How are they, what's the kind of range that's out there? And I wanted the specimens that we displayed to kind of walk the line in between art objects and specimens, objects of science. And so they are arranged in a particularly aesthetic ways. Some of them are groups of different plants, and some of them all always from these same kind of micro communities. Like they aren't mixed and matched that way across communities, but they are done with aesthetics in mind very much. Although there were moments where, like, Nadia, my student assistant, you know, I gave her a lot of freedom to kind of come up with some compositions and then check in with me before we glued them down. And she came up with some like really beautiful arrangements. But I kind of was like, you know, that's not really what that plant does though. Like that doesn't relate at all to the growth habit of the plant. So we kind of pulled it back. So we were kind of navigating this in between, I would say.
Yeah. Neat.
I have a question in the chat here from Arlene. She's asking, thank you for such an interesting session. I was wondering about the storage of the herbarium today. What are some of the challenges you face when preserving this type of natural material?
Well, you want to, the entire herbarium, we have 1.5 million specimens in various cabinets and a lot of cabinets. The types room is smaller. We have about 50,000 specimens in there. But there are a couple of, the enemies are moisture and humidity. So you try to keep constant temperature and humidity, low temperature if possible, which also has the added benefit of slowing down the little critters that would be crawling in to lay eggs on the specimens, which would chew them up. For short-term storage, that doesn't really show up much, but over the centuries, one of the pests is called cigarette beetles, and they probably started out in North Carolina and infected their way across the world, basically, or at least in North America. And so you want to keep those pests out. They used to spray some insecticides, but I think they kind of noticed the herbarium curators are not lasting as long as they should. So we don't do that anymore, but we do keep it cold. And we do integrated pest management where we put traps out, and if we find this evidence that insects have gotten in, we freeze everything. And surprisingly, when these things are dry, you might think that that would hurt them. You don't put your spinach in the freezer to preserve it, right? But once it's dry, it comes out looking exactly like it went in.
That's fascinating. Patricia, you have your hand up.
I just wondered, just like DNA with animals and fossils and stuff, these materials aren't that old, but yet they grew at a time when the environment was very different than it is now in terms of carbon and everything else. Has there been any interest or is there any use of these samples as scientific research in terms of, do the cell structures of these plants, are they something you can investigate?
Yes, it hasn't been done very much with the Lewis and Clark plants, but about a decade, two decades ago, Earl and I were involved with a project on this. A small amount of leaf material was taken from some specimens, and we didn't look at DNA, but a biogeochemist from the Carnegie Institute in Washington, D.C., basically incinerated them and looked at carbon isotopes coming off. Plants prefer a particular isotope of carbon from the atmosphere, which is now enriched by fossil fuel burning, because it's old plants being released back into the atmosphere. And so if there's more CO2 in the atmosphere, more of that carbon isotope is taken up. And you can backtrack that way and calculate what CO2 used to be in the atmosphere over the world, really, but specifically North America. And CO2 was lower back then, which is not too surprising. So we don't want to, grind up a whole lot of Lewis and Clark plant material just to get the obvious stuff. But one of the plants of tobacco was thought to be extinct, but found in 2013. And we'd taken a leaf from that new specimen and done a next-gen sequencing, DNA sequencing on it. We'd kind of like to see whether or not it's related to Lewis and Clark specimens. Now there's, I think on the cover page for the talk here, that's a tobacco plant. Or no, it's not tobacco, but somebody showed one there. I think Allie did. There's a lot of leaf material there, so we could justify taking a small leaf and getting DNA from it if we wanted to. But are the specimens we have from modern times, which were grown from seeds kept by Native Americans, provided to us, are they the same lineages that Lewis and Clark collected? That's a nice hypothesis to test. But they all have DNA in one form or another, just like all the 1.5 million plants do. So that's really an untapped resource for biochemistry and DNA. Yeah.
I have a question, and this connects to the workshop that is on our schedule for next month. And this is for all three panelists. What kind of advice would you share with people who are wanting to create their own herbaria at home in terms of anything from like compositional arrangements to range of species, and when an herbarium is deemed complete, if there is such a thing, and then also in terms of the kinds of stories that they want to be telling with these collections and preservations.
Yes, if you want to say like stories too, if you think about if you're collecting these plants, for what reason, right? Is there a special connection to you? Is there a special connection to Philadelphia? Whose story do you want to tell? And that's in a lot of things of why we collect, for what reason, and what's the narrative there. So I guess it's like thinking about, when I'm doing this, why am I doing this? What do I want to show? What do I want to display? I'll leave the more science related aspect to our two experts here.
Yeah. Gina, you want to go? Talk or have
Sure. Yeah, so I think I actually kind of latched onto the same thing that you did, Ali, which is that Nava, you used the word story. And I think that this is a really, the process of making a herbarium is a kind of, it can be thought of as a narrative practice. There's a nice blog post if people are thinking of doing this that I found. It shows 19th century herbaria made by students at Mount Holyoke College, which is a women's college in Western Mass here, where I actually used to teach. And at that time, it was part of their education was that they had to take a walk every day. Maybe it was like 2 miles or something, and they had to make herbaria, and so they all like made their own. It was like of their walks, and I think it was like a really nice encapsulation. So I think that that's just a really nice way to think about doing it. I think I'd also just say like consider the impact on the plants. Also, so to think about like not digging up a rare plant and putting the entire organism onto your herbarium sheet, and just to think of your act of collecting as part of an ecological practice, and to think about it's pretty easy to take a lot of specimens without doing a lot of harm to plants, but to consider that and maybe seek advice from knowledgeable people before you start collecting. That's all. Thanks.
I guess I'd add for the specimens that we have, the reason we know they're Lewis and Clark is it has on them the notations from Lewis and other people who added later before the memory faded of where it was collected, the date it was collected, and sometimes the identification, although some were new species at the time. And sometimes a note. Lewis did note down sometimes whether antelope were eating some plant or whether the Native Americans used it for wounds, things like that. Not a whole lot of things, but that was more in the journals than on the herbarium sheet itself. But so you might write a few things down like that and think about what story you want to tell, whether this is your backyard herbarium. So maybe your grandkids or great grandkids will be living in the same house. That doesn't happen often, but maybe they'll have a garden. But if you keep getting things from Primex, you'll go get the same thing. But if it's a native plants type thing and you're collecting and you need permits in many cases nowadays, not so much in some places, but note where it is, and your phone will give you a geographic coordinates, latitude and longitude, which Lewis and Clark had to do all sorts of elaborate things to finding out where they were, and they didn't put any of the information on the plants at the time. So you can add that, and also take an iNaturalist picture, and then you'll geotag the specimen before it was collected. So there's lots of things you could do that way. But think what you're going to want to do. You might put it in a book if you're going to keep it for yourself. I see no problem with that. It's probably stores better. If you want to give it to an herbarium someday, have all that scientific data I mentioned of what, where, and when it was collected, and maybe you can't identify it, but somebody can, because then that gives it value to it, and it might be that the herbarium can use it. But if it's just a plant that's pressed between the pages of your copy of Shakespeare or the Bible, it might not be all that scientifically useful.
I have a question for Gina, and feel free to also chime in, Ali and Rick. Actually, it does it directly to you, Ali, as well. When we were just viewing the current exhibition, Drawn in Nature, we were looking at some of the site maps that were hand-drawn, and Gina also drew a site map for her project. And I just wanted to hear a little bit about that process and what that looks like, especially since that correlates to the herbarium.
Thanks for that question, Katie. Yeah, I loved seeing the maps in the exhibit, the APS. That was so fun. There's something so appealing about hand-drawn maps. As Nava knows, I kind of dragged my feet on producing our map. I think I spent too many years studying drawing, and so I got myself into a crisis of overthinking. But I got over myself, and then I had a lot of fun doing it. We tried to use our map, Kate and I, to represent kind of like the different ways of knowing that are involved in the project. So some of it was like empirical observation, like this type of thing is growing here. There's lots of birch trees over here, you know, and then some of it is memories. Like this is where I saw the barred owl that morning in 2020 when I didn't have my binoculars, or this is, you know, where Kate stepped through the ice, which was a memorable moment in our project. So there's a kind of mixture, you know, we have had all these class visits and public engagements at the site. And so the site is like a dense place for us of observation and information in many organisms and this complex ecosystem, and also memories and experiences and different kinds of learning. And so the map relates to that. And in order to produce that, we did an exercise where I made like small versions that were mostly empty on smaller pieces of paper. And then we went out to the site together, and I was like, okay, I'm going to set a timer for a half an hour, and we're going to just walk around the site separately and mark everything that we think is important and can remember. And it was like 15 degrees in January. It was really cold. So we're like out there with our freezing hands, writing on these maps. And then we kind of came back together and we walked each other through our own maps, which sort of brought together more memories. And then I took both of our maps and I made a mashup of them into the big hand-drawn map. So there was a little bit of a process involved. The hand-drawn map will be in the exhibition catalog, which will be coming out pretty soon. So yeah, come check out the catalog.
Yeah, maps are, especially in the exhibition, they're really interesting too, because they can tell multiple stories, especially in the 1700s, 1800s. Maps were used for a variety of purposes. One, to share information, right? Like where they found something, but also to show where these species might come from, what lives there, but also to show too of like, there's also a commercial aspect to many of these naturalists' ventures in the 1700s and 1800s, right? They're showing what resources are out there, what species are out there, and also what land is out there. So it's kind of this twofold idea of look at these species we're discovering, and it's also, but like, look at all this land as well. And so it's these naturalists both have a positive and kind of a negative impact at times for their actions and what the information they're sharing out. So but yeah, I won't go on anymore after that, but come find me in the exhibition. I'm happy to chat more.
Do we have any other questions? No? Great. Well, we will be giving you back 6 minutes of your day. So I just want to thank Allie, Rick, and Gina for so kindly bringing all of this wonderful information to us today. I learned a lot today. I hope you all did too. Definitely check out all of our organizations and follow us on Instagram. We do have an herbarium workshop coming up, so check our website. It's June 1st at Glen Ford on their grounds. So we will be making our own, you'll be able to make your very own plant press and take that home with you. So you can keep exploring nature on your own terms. Check out our current exhibition to understand a tree and see Gina's work. And this talk will be available in the next couple of days. So check our website and YouTube, and also our partners with the APS and the Academy of Natural Sciences. And yeah, thank you. Have a wonderful day. Enjoy this cooler weather that we're experiencing here in Philadelphia after yesterday feeling like we were in Louisiana. So,
True.
yeah, it did. Very humid here. So thank you so much, everyone. Have a wonderful day, and we look forward to seeing you back here for our next event. All right, take care. Bye-bye.
Thank you.
Thank you.
Thank you so much. Really, really wonderful. Thank you.
Bye-bye.
Great.